Okay, I want to thank everybody for joining us for today's live webinar with Astrix and AgiLab. Today's topic is Think Faster, Decide Better, How AI and Lab Informatics is Transforming Scientific Decision Making. Today's session is presented and sponsored by Astrix and will be actually presented by AgiLab. And before we get into today's session, I just want to take a moment. If you haven't been on any Astrix webinars before, I'd like to take a moment and introduce exactly who we are. So we can go to the next slide, Anahita. And just a brief kind of introduction as to who Astrix is. We're a consulting firm that specializes in strategic and technical services, processes, and we are a technology agnostic provider, meaning our approach is that we don't espouse one technology, we espouse the process of helping companies choose the right technologies for their R&D, lab, clinical, etc. We've been doing this for over 30 years. We work globally. We have most of our offices are in the United States. 90% of the employees that currently work for Astrix hold advanced scientific degrees. We are very much a science-based company helping other science-based companies. And again, we've been serving the scientific community now since 1995. So that's a quick overview of who Astrix is. Again, strategic and technical consulting firm specializing in life sciences. With that, I want to take a moment and introduce our speaker for today. So Anahita Lakowski is going to be the speaker and the presenter. Anahita, before we do the poll, why don't you take a couple of minutes and tell the audience a little bit about yourself?
Yeah, absolutely. Thank you for that. Hi, everyone. Thank you for joining us today. My name is Anita Laskowski. I'm a product specialist at AgiLab. My background is that I studied a master's in chemistry, and I've been working in the lab informatics industry for over 15 years. During that time, I've been involved in over 200 projects, helping various industries in their digital transformation, from pharma to biotech, chemical manufacturing, as a validation consultant, LIMS consultant, service delivery manager, solutions architect, and as of the last few years, I've been a product specialist. And just a little bit about AgiLab. We are a laboratory informatics company. We're built on a low-code modern architecture, and we have LIMS, ELN, LES, and EBR on a single connected environment. We help industries from R&D all the way through to QC, manufacturing, and our goal is to empower scientists and lab managers to make faster and smarter decisions. And that's what we're going to do today. So I think we're going to start with our quick poll just to see where everyone is in their AI journey, because this session is going to be around AI in the labs. So if
Yeah, you should be seeing that on your screen right now for the attendees, what AI ML maturity stage is your lab at. So we'll leave this up for about 30 seconds, 45 seconds or so, give everybody a chance to respond. I want to encourage everybody to just take a moment and make the most logical selection as to where you are at currently. And then what we'll do is we will publish the results in just a moment so everybody can kind of see where you're at. It's not going to show any individual responses. It shows a summary of the responses.
perfect.
We'll give that another, we'll give that another sec.
No worries. I'll just open the agenda slide.
All right, we're going to wrap that up in about 10 seconds here. So if you haven't responded, I'd encourage you to do so. Okay. Thank you for participating. I appreciate that. Let me go ahead and share those results for everybody. So you see those up on the screen. So it looks like coming in number one, we've deployed AI in some workflows, but want to do more at 44%. We've got, we are still exploring what AI could mean for us at 33%. We have discussed it internally, but haven't taken any concrete steps forward. Sorry, that looks like that question cut off at 22%. So those were the three main categories that folks had responded to. Appreciate your participation. Great. Well, Anahita, the floor is now yours. I'll come back on at the end and help out with any questions. And again, I want to encourage everybody, please ask as many questions as you want as we go through the session.
Perfect. Thank you so much. Okay, so over the next 45 minutes, I want to talk about something that I'm genuinely excited about after working for 15 plus years in this industry. And it's a shift that's happening in the lab right now. but it's also one that I feel like is quite misunderstood, and that's AI in the laboratories. So it's not the kind of robotic version that's going to come after your jobs. It's the real practical, what does it look like on a Tuesday morning for your scientists kind of version. So with that, we're going to talk about why AI, why is it relevant now, before we move on to some AI use cases, a short demo, and then we're going to look at some slides on implementation realities and also what AI looks like in regulated labs. So first slide with this, why AI? Basically, the short answer is everything is more now. Everything is bigger now. Look at data these days, right? So we have 10%, 10 times more data generated than we did 5 years ago. 40% more of the scientist's time is being spent because of that on routine data reviews. 60% of that 60% of the delays are because of data insight. Now, this is actually, these figures are not from any published studies. These are actually from our experience speaking with our customers, our potential leads, and just talking about what are your general pain points. And it all comes back to this. But if you're working in the lab already, I think these figures were either not be too far off, or they might be even a bit conservative for you. And the problem with the modern lab is that it's not short of data, but it's short of time and clarity to act on data. Because really data is everywhere, but insight is where the bottleneck comes in. And that's where AI can help. So as the review queues grow longer, the results come in faster, then they can be processed. The scientists should be making high value decisions. But what we find is they actually end up spending a significant amount of their day working on admin work, let's say, or chasing signatures for data review. And that's a decision bottleneck. So, and as it gets, and labs generate more data, they run more assays, there's going to be more and more data and more of a bottleneck here. And this is where AI can actually solve the problem when it's designed properly. So before we get into the use cases, I just want to set the tone for this session, which is AI is not here to replace the scientist. I know it sounds like something reassuring that people just say, but I genuinely mean it. And it's not just philosophically, because the way at AgiLab we view AI capabilities is that the scientist is always at the center of it and is always in the loop. So, and this is why this matters a lot, because scientific judgment shouldn't be looked at as just a process step. It's a contextual, irreplaceable expertise that you need. So the scientist is the one who looks at a flagged result that could be flagged by AI and say, yeah, that's fine, because I know that this batch had a temperature excursion last Tuesday, and they make a final call on that. What AI can do is take away all the noise and surface the signal. So it can flag the anomaly before it gets buried, categorize the cues so that the scientist's attention goes to where it needs to go. And every AI output, the way we look at it, is a prompt for scientific review. So it's never a final verdict. It's basically the scientist translates it, contextualizes it, and then decides what to do. And that's why we have this AI handles volume and scientist makes the call. And that's the philosophy behind all of the use cases that I'm about to show you. So use case number one is intelligent anomaly detection. And just to set the scene for you here as well, I'm going to start off with a question for you to think about, which is, have you ever caught out an out of spec result just a little bit too late because it was buried in a large data review queue? Because this is something we have asked our laboratory teams, and the answer is almost always, yeah, we have. And the following question is, okay, what did it cost you? How much retest, investigations, and even potentially a batch failure? And all of it downstream from one result that could have been caught earlier. And here's the traditional approach, which is the scientists can look at the data set and look for kind of any out of trend. Because as you know, if most of you will have a limb system in one form or the other, it's absolutely possible to flag out of trend, out of spec, and see, yep, this is flagged as red. But what that flag doesn't tell you is, for example, that reagent has a lot history, because every data point is treated the same. It's that this pH is out of spec within this range. And then it's up to the scientists to then take the time, do all the relevant searches, look at the equipment it was tested with, look at the reagents to make the final decision. And all of that takes time. So what we've done here with AgiLab is we've offered additional data by factoring in batch history, reagent lots, even analyst patterns, and environmental variables. So the difference is going to be that when something does deviate from something that's expected, it gets flagged immediately, but this time with context. Okay, so it's not just a red flag. It's here's something that went wrong, and here are all the details with it, and also some similar events that happened in the past. And crucially, the flag is a prompt. It's not a final verdict, but the scientists will review it, they will apply their expertise, and then decide what to do with it. So it's just all about making it easier for the scientists. And the outcome being there's just fewer anomalies reaching late stage review, faster investigations, and the scientists spending their time and attention on what actually matters. Second one is predictive analysis. This is probably the one I get asked most about. It really does have the most amount of interest. And the idea is simple. So instead of waiting for a full analytical run to complete before you know a sample will pass or fail, what you can have is an early detection while it's still in progress. And think about where this matters the most, which is biologics, long-term cycle QC assays, complex multi-step workflows, where a sample failed at the end means there's hours of lost runtime that are wasted materials, delayed batch decisions. And really in those environments, if you know early, even based on probability, it can be very valuable. And here's how it works. With AgiLab, we would train predictive models on historical run data. So it would be your historical run data, and the model learns the pattern that generates either a passing or a failing result. And when the sample is running, the model generates a probability signal in real time, not on a result. It's just a signal that's saying this sample is possibly trending towards a failure. So the scientist kind of knows before the run. Now, this is something where I want to be careful, because I don't want anybody to think that what we're saying is that you can replace actual results with probability, because especially in regulated environments, the predictive signal is just complementing the actual analytical result. It doesn't, you still have to have the run, and you still need to have the results, and the scientist still needs to review it. But what changes here is that you can make earlier decisions about prioritization of your resource allocation, or whether to get involved mid-process, but this is going to be based on the scientist's judgment. And the outcome of this is faster pipeline decisions, less time waiting, and laboratory that's basically ahead of its data rather than always chasing it. In the next slide, this is just the training model that we have. So algorithm is constantly improving itself. The more data you have, the more it can run on that model. The more repeating the same processes, you're reducing the number of the errors that you can get from the algorithm. And just to also tell you, sample predictive analysis is not the only thing you can use this model for. You can use it for stability studies, QC batches, equipment maintenance, and even reagent inventories. Because the concept is the same. The concept is that you train the model with your data for a behavior that's going to be expected by a probability. Use case number 3 is actually a fairly recent one, and it's predictive resource scheduling. We had this brought to us by a customer recently as a huge pain point and something that was costing them a lot of time and money in the labs, and it was resource scheduling. They told us what they had was several staff who were spending significant amount of their time just going through spreadsheets, whiteboards, booking systems, and SharePoints just to create a schedule for their labs. So based on the staff they had coordinated, on their equipment, and on their analysis. And what we did based on the data they had is putting all this data together and allowing the information, having it in one place and allowing the AI to do the pattern recognition across all the data sources. So sample queues and priorities, then we had instrument availability, current performance statuses, staff rotas, competency records, historical throughput of the data, and all together in real time, we've generated an optimized daily resource planning. So if you look on the right-hand side, what this can look like in your lab is you have your instruments, you have your staff, and your analysis. Here we have 5 instruments, 5 staff, and 5 analysis. On line number 1, HPLC is ready. It's calibrated and qualified, ready to go. Then we have Dr. Chen, who is also available and trained to run assay A and is scheduled for 9 a.m. All good. Line number 2, there's a drift alert in HPLC number 2. And also S. Patel is in calibration training. So they can't actually run this, which means assay B is potentially going to be delayed. But what the AI has done by itself is, if you look at line number 3, it's taken the GCMS and another member of staff who is also trained to run assay B and rerouted it to them. And now the thing to remember here, this is all just what the AI decides, and the lab manager needs to have the bottom line. They can look at that and say, well, actually, no, he can't do that. This is basically what I meant in the beginning, that we're just trying to make it easier by providing the right data for staff to make the decisions. And then at the bottom, you see it gives you a full kind of report of insight of HPLC number 2 has a drift, S. Patel is unavailable, assay B is rerouted, GCMS is with M. Costa, and everything is accounted for. Okay, so the lab manager can then come in and review this, but there could be, for example, let's say a customer priority that will change the order of this, or a training opportunity for a junior analyst, and they can say, okay, let's reroute this. So the AI has no way of knowing that, and that's why you still need somebody to work with the AI. But it's, in this case, it's just much faster. So the outcome is straightforward, but it's genuinely significant. Like, this might look like something quite small, but it's really saving a lot of time and effort. So there's higher throughput with the same resources, because they're not losing time on these kind of avoidable conflicts. And there's also better utilization of your staff in the lab, because qualified analysts should be doing the job that they're trained to do, rather than any kind of admin work. So what I want to mention as well, in a GXP environment, assigning the right qualified staff to the right to the right analysis, this is something actually that's a compliance requirement. So you're actually having another tick off there as well, because everything is audited as we go through. So just because AI does it, it doesn't mean it's not audited. Everything is also audited, even though AI is running it. Then we have use case number 4, which is semantic search. So semantic search is probably, I would say, the simplest one, but the most effective. I just from as far as I know, how much time it can take to bring the data back from your LIMS that you already have. So this allows a scientist to search across their LIMS records, SOPs, deviation reports, historical results using natural language queries, just instead of having rigid filters. Now, depending on which LIMS system you're on, either you have to know kind of database field names, or you have to know exactly which module to search when you're looking for a specific data. And just for anybody in the audience who doesn't know what semantic search is, somebody explained it to me in this way, and I thought it was kind of fun, but also easy to understand. And it was, if you're looking for a banana, you can type in, show me the yellow fruit. And what it will do is it will show you a mango, a lemon, a star fruit, and a banana. So that's really like the simplest decision, because the definition, because it actually understands your intent behind your query. So it doesn't have to be exact, but it will understand what you're asking. So you can see on the right hand side there how the old way was. So you're looking for a specific module, specific field, and you're looking even with the wildcard search, which is the percentage. With semantic search, you can just say, show me all the deviations from temperature excursions on instrument number 4. And then that's the result you will get. So even, this is just going to make life so much easier for scientists that can just type in batches with yield issues in Q3 involving reagent lot X, instead of having to look at every single batch individually or every single reagent individually to see where they've been used. So even with the exact words used in the query, sometimes they don't appear, and the search engine matches meaning with instead of syntax here. So why this is meaningful is laboratory data is voluminous, it's vast, and often it's siloed data. So scientists tend to waste a lot of their time navigating rigid search interfaces, switching between modules, or just simply not finding historical information that they know exists somewhere in the system. And this is only if it's connected. Some data is even on paper. But what semantic search does, it makes institutional knowledge accessible in seconds rather than minutes or even hours, to be honest. And the scientist's role is that the scientist asks the question in their own words, the system surfaces the most relevant records, they review it and decide what is useful. So this is really, if you think about it, no different from a Google search. But the only difference here is that this is based on your laboratory's data, okay? Because if you think about Google, you don't act on every search that you make, you act on things that you know, okay? So, and the outcome is that faster access to relevant historical data, especially during an investigation, deviation, or even an audit preparation, because this is something that can take a long time. An auditor is asking for a specific deviation. Where did it come from? Instead of having to run through countless audit logs, you can just ask the system and it will bring everything back to you. And it leads to better informed decisions because scientists are actually finding the context they need when they need it. Now, one thing that's quite important to take away from semantic search, and honestly, AI in general as we go, you'll see in the other slides, is that fair is no longer the goal. So when you're implementing a lab informatics system, or when you're thinking about lab digitization, fair is not the final goal. AI ready is going to be your final goal. You will see this trend as we go. And it's something that I truly believe, even if you haven't thought about AI or you're possibly thinking about it, AI ready data is so much more important nowadays. And with that, I just wanted to quickly show you a video just to show you how simple, one sec, how simple semantic search is. So I'm just going to share this. I think that might be a bit blurry due to my internet. Is that, oh, there we go. It's here now. So here you can see, for example, you can say, give me 3 batches. And as you can see, he's even made a spelling mistake while he's asking for the batches in detail. But what it does, because it's semantic, it understands what you're asking. So we'll just go ahead and show you those batches. Another question that was in the beginning, it was actually asking the system how to make a fraction. So you can even ask the system, it can act like a system reference guide. So you can say, show me how to create a batch in your system, and it will go ahead and show different steps, or it can go ahead and actually make that for you. So here he's asking, can you give me batch 001 and 002 in a compare table? So if you need to ever compare your data, it can also do that for you and just display the data. So it's nothing, if you think about it, super high tech. It's really simple because you actually have that data in your lab. It's just a case of having the data that you can really use. So I'm going to go back to this. We had the demo video. So now let's talk about the actual implementation realities, because, you know, all this is great, it looks good, but what does it mean in a real life lab? So something you probably already know is that AI is not going to just land in your lab and start working. There's things that need to be in place, and it's probably not going to be a smooth ride to begin with. So there are 4 things you need to consider before jumping in. And number 1 is always data readiness. So AI is really as good as the data it learns from. So if your LIMS is messy, you have inconsistent or poorly governed data, or in separate systems, even worse on paper, that's where you should be starting. Don't even look at the model, don't even look at the use case. You should be looking at clean, structured, traceable data, because that's going to be the foundation for everything. It's not high tech, it's not fancy, but it's absolutely a non-negotiable. The second one, if you are in a validated environment, is making sure that everything is validated at every step. So you need to understand the intended uses, the performance qualifications, change control, and this is something you need to be thinking to build from day one, because if it's an afterthought, it's going to take a lot of time and effort. Just to be clear, this is not a small task. Then the third one is integration. So for those of you who are actually on a legacy system, the good news is that you don't need to have the whole infrastructure in place to have data, to have AI. All you need to do is have the data. And what you can just make sure is that you have APIs to connect to your system to pull the data to enable the AI. And this way you can do a phased, low risk adoption path that's kind of entirely achievable. You don't have to do everything at once. So if you do have a legacy system, you can still have an AI enabled lab. It's just going to have to be at a phased decision making. Then we have finally the change management, which is potentially one of the biggest risks. It's not technical, but you need to make sure that the system you're selecting, your scientists are behind it, they trust it, and they know that they can rely on this system. So there needs to be transparent outputs, clear explanations, and also gradual rollouts is what builds that trust. So this brings me to the question I know that you've probably been thinking about, which is what does AI actually look like in a GXP regulated environment? So there are three pillars to this, and it's not aspirational, it's architecture, and you have to build around them. So the first is that your scientists stay in control, so that AI is always the prompt, not the final verdict, and your workflow steps don't actually close automatically without the scientist making the final decision, the final approval, the electronic signatures, all of that will still be by the scientist. So it's not a compromise, it's a decision principle. Number two is that your data is actually secure because it's not an open AI connection. It's AI based on your data. You're not sharing data with anyone else. It's just your data. So everything is private, either on your internal private servers or on a SaaS company that you've selected. Your data should never be shared. It should only be your data. So it would be our models, your data. Number three is data integrity at every step. So basically, the AI generated flags, predictions, and summaries are all logged in audit trails. So just because AI does something, it doesn't mean it won't be traceable. So nothing happens in the background with no record. The audit trail covers complete decision chain from raw data all the way through to the final approval by the scientist. And regulatory frameworks like 21 CFR Part 11 and GXP, they don't block AI. They basically just define how you're going to be implementing it properly. And when you start with those principles rather than adding them later as you go, then you know that you have responsible AI in regulated labs. So just to zoom out from what we've been looking for, because what we've been looking for are things like anomaly detection, predictive modeling, semantic search. These are, they're great, but they're first generation of AI in labs. They're definitely valuable and they're definitely real. But I think the direction of travel is towards something much more ambitious. And these three things are going to define, I think, where things are going. And number one is autonomous lab workflows. So routine processes like sample scheduling, instrument allocation, and so on, they will be handled by AI. So not to, again, remove the scientists from the lab, but they will just free them entirely of these sort of administrative works. Then we have closed loop experiments. So it's that AI doesn't just analyze the results for you, but it will actually propose the next steps. So based on these outcomes and also historical performance and scientific literature, this is going to be the next step, right? So it's going to be acting as a genuine decision partner and not just a data processor. Then we have real-time intelligence. So this is continuous live monitoring across all lab workflows with predictive alerts, trend forecasting, and dynamic resource allocations before problems actually arise. The lab informatics industry is building towards this future. I know that there's a lot of skepticism, so I understand that fully. And I think the bottom line is that this is where we're headed, and you know, the principles are going to remain the same. We can start quite small with, you know, predictive analysis or things like semantic search. But the future of the lab science is not without science, but it's definitely with more data and allowing more tools for the scientists to make the final decisions. So with that, and before we stop with questions, I want to talk about the things to think about if you're actually either thinking about implementing AI or thinking about implementing more AI. Because everything we've talked about today, the decision bottleneck, the use cases, responsible AI, it all points to something bigger, and it's something that all laboratory leaders and lab managers and scientists and anybody in position of influence should be thinking about right now. And it's that you're building your kind of the lab of the future today. It's not in 5 years. So even if you have absolutely no plans for AI, you need to be thinking about AI ready data. Okay. So not when AI is mature enough or more regulated, but the decisions, all the decisions you make right now about your systems you choose, the data you collect, the infrastructure you invest in, these decisions are not kind of without AI in place. You need to think about that because they're either closing the door for a kind of more high-tech lab or opening them. So there's a few questions to think about for you guys. It's not to answer today, but to go away and maybe really think, is your lab process future proof? That's one of them. So to answer that, you need to think about, is your data AI ready? Okay, so this is a really foundational question you should be asking yourself and your team, because AI doesn't create value out of thin air. It's going to learn from your data. So the more quality, structure, and governance your laboratory data has, the more you're going to have a better chance at deploying a quality AI system. So if your data is siloed, inconsistently structured, poorly attributed, or living in spreadsheets or outside of your LIMS, this is not just data integrity risk today. This is actually an AI readiness problem for the future. So every decision you make, you should be evaluating, does this generate more traceable, clean, structured data that I can use in the future? And if the answer is no, you should rethink that. Then the second one is, are your lab tools future proof, right? So when you're evaluating possibly a new platform or assessing the one you have already, the question shouldn't be, does it do what we need today? It should be, does it make our scientists better at their jobs? So that's basically a trap that many organizations fall into because they're just digitizing the same old workflows. So they're just taking paper processes, putting them into software and saying, okay, transformation done. But that's not transformation. That's digitization. So the real transformation asks, how can this system surface better information, reduce cognitive burden, and give my scientists the clarity they actually need to make faster decisions and with more confidence? So that's the bar I think every laboratory should be held to. Third one is, can your platform grow with AI? And that's something, AI in the lab is not a feature you can buy or something that you can tick off. It's a capability that it needs to evolve, and it's evolving fast. So the platforms need to serve you well over the next decade with things like open architectures, with API connectivities, backed by vendors who are actually genuinely actively investing in intelligent automation. And when you're in procurement and you're having these conversations, you should be asking these vendors about their AI roadmap. And finally, is the scientist still at the center? So this one I feel most strongly about, and it's that the labs that will lead in this in the next decade are the ones that actually augment their scientists the best. So technology should be amplifying the human experience and not replacing it. So don't bypass your scientists. What you want to do is just the scientist's judgment, intuition, and domain knowledge should be your most valuable asset. So then every decision you make is this actually adding more value to my asset? And if not, again, you should be rethinking that decision. And with that, we're exactly at our 45 minutes. So I will move on to the Q&A part. Thank you so much for joining us and for your attention today.
Great. Thank you so much. Great presentation. I'm going to go ahead and stop the recording now. We typically don't record the questions, so let me get that stopped.